All right, so good morning, everyone. So today we are going to continue chapter 4. We are going to talk about the functions of Sertoli cells. Now, last week we talked about the structure of the Sertoli cells. We mentioned the apical and lateral processes, and we talked about the importance of tight junctions that exist between the Sertoli cells, as well as the other types of junctions that exist
راح الصوت شي
No.
So,
Can you hear me now? Can you
yes,
hear me now? Okay.
yes.
So I was saying that we talked also about the tight junctions that exist between the Sertoli cells as well as the junctions, the desmosome and the gap junctions that exist between Sertoli cells themselves and Sertoli and germ cells. And also we talked that the Sertoli cells contain two types of receptors, a membrane receptor in order to respond to the FSH molecule, as well as a nuclear receptor. So it's an intracellular receptor which is to respond to the testosterone. We will see this today in details. Now, the table 4.1 inside your books will mention all the functions of Sertoli cells. So we are going to go into each one of these functions. We start with the first one, which is the support function of the Sertoli cells. Now, as you can see here in front of you, so we have the support. What is the importance of the function of support given by the Sertoli cells? We mentioned before today, the weeks before, that the Sertoli cells are the nursing or the nurturing cells or the mother cells for the germ cells, because these Sertoli cells will provide nutrients, will provide molecules that are important for the development of the germ cells and for spermatogenesis. Now, also very important function is the support, whereby the Sertoli cells, as we mentioned, they have tight junctions to divide, we said, the compartments into luminal and basal compartment. So these supporting cells can modify the junctions in order to allow the primary spermatocytes that are in the pre-lactotin stage to pass to the luminal compartment. So they will modify the junctions in order to allow the displacement of the germinal cells. So they are supporting for these cells. They keep them in the basal compartment to provide them with the nutrients, and they can allow them at a certain moment to move to the luminal compartment to be exposed to other types of molecules. Now, also these Sertoli cells, they allow the exchange of metabolic substrates and waste between the spermatogenic cells and the blood. The second function is the phagocytosis. Also, we mentioned that during the maturation of the spermatid, when the spermatid is acquiring a cytodifferentiation to become a sperm cell, it will lose a big part of its cytoplasm in the form of a residual body. These residual bodies that are removed by the Sertoli cells, they are phagocytosed and resolved by them. And any cell that fail also to differentiate will be at the same time phagocytosed and got rid of by the Sertoli cells. The third function is spermiation. It's very important to release the sperms from the apical part. So we saw also in the figures before that the spermatids are attached to the Sertoli cells, and because of their detachment from these Sertoli cells, these sperm cells will find themselves released in the lumen of the seminiferous tubule. The number 4 function is the secretion, and when we talk about secretion, there are molecules that are secreted to act in an endocrine way, or exocrine, or paracrine. Examples of secretions include the inhibin. Now, inhibin is secreted by Sertoli cells to inhibit the proliferation of the spermatogonia, as well as to inhibit the secretion of FSH by acting in a negative feedback. We will see this today. The activin, also inhibin and activins, are two types of cytokines that we saw in the reproductive messengers chapter. Activin acts oppositely to the inhibin. It acts to stimulate the proliferation of spermatogonial cells and to stimulate the secretion of FSH. The third type of secretion is done by the Sertoli cells, which is the interleukin-1 and interleukin-6. These two molecules, they act differently. Interleukin-1 stimulates the replication of meiotic and mitotic DNA. However, interleukin-6 inhibits the replication of meiotic and mitotic DNA. So before mitosis and before meiosis, the cell should replicate its DNA. So these two molecules will be either stimulating or inhibiting the replication of meiotic and mitotic DNA. The fourth molecule is the growth factors, IGF-1 and TGF-beta. So IGF-1, which is insulin-like growth factor, and TGF-beta is the transforming growth factor, are two important growth factors that are very important to control division and differentiation of germ cells. A fifth molecule that is also important and secreted by Sertoli cell is the plasminogen activator. What is plasminogen activator? As its name, it will activate plasminogen. Now, plasminogen, when it is secreted in a form that is inactive. And thus, when Sertoli cell secretes plasminogen activator, this plasminogen activator will be able to convert this plasminogen, which is inactive enzyme, into an active plasmin enzyme. And this plasmin, which is a protease, is very important because it's this one that will be degrading the inter-Sertoli tight junctions. So the tight junctions that are present between the Sertoli cells will be degraded, and this thus will allow the primary spermatocytes that are in the pre-leptotin stage to pass from the basal compartment to the abdominal compartment once they want to enter prophase 1. Okay, so they pass from the basal to the central abdominal compartment. And also this plasmin is important to degrade the desmosome and the gap junctions that are present between the Sertoli cells and the germ cells, and also will allow the resorption of the body, the residual body, once it is formed. Other molecules include the transferrin and ceruloplasmin. These two molecules, transferrin and ceruloplasmin, are carriers and transporters of so transferrin for the iron and ceruloplasmin for the copper. So they will deliver these two types of minerals to the germ cells by transporting them. Also, these two molecules are secreted by Sertoli cells. So all of these molecules that I'm talking about now are secreted by the Sertoli cells. The androgen binding protein is a protein that is important to transport and concentrate testosterone in the tubular lumen. Now, testosterone is secreted, is produced by the Leydig cells. Once it is delivered to the Sertoli cells, it can pass through the membrane, since it is a lipid, it is a steroid, so it can pass through the membrane of the Sertoli cells, and then it can pass to the lumen of the tubular, and where it binds to the androgen binding protein and gets concentrated in the tubular lumen, as well as later in the epididymis. The anti-Mullerian hormone, which we mentioned when we talked about the gonadal differentiation and the formation of the Wolffian ducts that were developed due to testosterone, we mentioned that the Mullerian ducts, which are the female ducts, were regressed due to the effect of the anti-Mullerian hormone. And we said that at that time that it has so many other names like Mullerian inhibiting substance, like inhibiting factor or hormone. This molecule, the anti-Mullerian hormone, is secreted by the Sertoli cells and is responsible for the regression of the Mullerian ducts. Now, these are the different secretions that are done by Sertoli cells. Now, the fifth function of Sertoli cells is that it provides what we talk about as blood-testis barrier. I already mentioned this last week, but today I'm going to go more into its detail, showing you in this figure. So you have here the basement membrane, and you have the interstitial space where you have the blood vessels, you have the Leydig cells that are present in the interstitial media. Now, this one here, this interstitial space, which contains the blood vessels, can be exposed to so many molecules, so many substances in this basal compartment. And these substances can pass to the basal part of the seminiferous tubule by either diffusion or by some of the transporters. Now, to prevent these molecules that are present at the level of the blood to reach into the lumen of the seminiferous tubule, these tight junctions will make sure that we have what you call as blood-testis barrier. Blood, because we are talking about the blood in the basal, toward the interstitial space, which is in contact with the basal compartment, and the testes, which we mentioned here, the lumen compartment where the sperm cells are present. Therefore, this blood-testis barrier is created because of the presence of these tight junctions that exist between the Sertoli cells toward the basal compartment. Okay, and this polarization will make sure that we have two different compartments, and the composition of the lumen of the seminiferous tubule will be different from that of the blood composition. The basal compartment here, where I mean this one, so this is the basal compartment, so all the seminiferous tubule, including the germ cells and the Sertoli cells from this tight junctions toward the basement membrane, will contain all the substances that are needed to for mitosis and for the proliferation of the germinal cells and their development till the prelactotin stage. However, every molecule or substance that is present in the abdominal compartment, which is after the tight junctions and in the lumen compartment, they are the substances that are needed for meiosis to be accomplished. And how is this maintained? How are these substances only allowed to stay in this part and not move to this part unless in a controlled way, because of the presence of these tight junctions that divided the seminiferous tubule into two compartments. Now, this barrier is functional, as we mentioned, it is functional after puberty. It's a protective immune barrier, so it will prevent any blood-borne substance to pass from the blood and reach to the sperm. For example, antibodies that are circulating in the blood, we prevent them from going to the lumen and affect the sperm cells. Any toxic, any bacteria, anything that is toxic or pathogenic or can cause a defect for the sperms in the lumen, all these molecules will be prevented from passing from the blood to the lumen where the sperm cells are present. Now, this barrier, therefore, is a barrier between the between the interstitial space containing the blood vessels. Of course, you have here the myoid cells that are the peritubular cells, which are the contractile cells in the basement membrane. There are the fibroblasts. There are the collagen fibers, the Leydig cells, and the blood vessels. All of these are part of this basal compartment or basal part. In addition, of course, to the Sertoli cells and the germ cells. And in the lumen, you have all the cells that are undergoing cytodifferentiation to reach to the formation of the sperm cell. Now, how are the molecules controlled? Now, you all know that here you have this big one here is the Sertoli cell. Now, these Sertoli cells, any molecule that needs to pass from this compartment to reach to the lumen, it needs to pass through the membrane of the Sertoli cells. So this molecule should be first a highly hydrophobic, like steroids, for example. So any molecule that is large, hydrophilic cannot pass. Okay, it cannot pass the membrane to reach to this part. And of course, one would think that they can pass between the cells. This is not true, because when they reach to the tight junctions, they will be stuck here and they will not be able to cross the tight junctions to reach to this place. So the only way they need to cross from this part of the section of the testes to the lumen is to pass through the Sertoli cells, and this is limited to small molecules and will prevent any molecule that is large and hydrophilic from crossing to the lumen of the seminiferous tubule. Okay, so only steroids can pass intratubularly, which means from the outside of the tube into the inside of the tube, passing through the Sertoli cells. Now we move to the Leydig cells. Now the Leydig cells are structurally and functionally different from the Sertoli cells. When we talk about the Leydig cells, first they are polygonal shaped. So you can see here, so they are polygonal shaped. And they have a nucleus that contains so many nuclei. Their cytoplasm is rich in smooth endoplasmic reticulum, mitochondria, and liposomes. Of course, because they are the site of synthesis of the steroids, and principal role of this cells are the synthesis of testosterone. They make 95% of the plasma testosterone is secreted by the Leydig cells. Now, these Leydig cells can be either present as isolated cells or clustered around the blood and lymphatic capillaries, of course, in the interstitial media. And they synthesize the testicular androgens, and particularly the testosterone. Also, they can synthesize testosterone from cholesterol, if we get the cholesterol from the diet, or de novo when we synthesize it from the acetate. Now, the testosterone is the major androgen that is produced by Leydig cells. So you can see these are the Leydig cells. So this is the basement membrane, and this is the inside of the seminiferous tubule. You have the Sertoli cell, this big cell that is a polarized cell. You have the lumen here, and you have the basal membrane. Sertoli's Leydig cells are towards the interstitial media. They secrete testosterone as the major androgen. Now, this testosterone can be bioconverged, which means it can be transformed into a more active or more potent androgen, which is dihydrotestosterone, via the enzyme reductase, or into also a powerful estrogen, which is estradiol, via the activity of aromatase. Okay, so this testosterone is produced by the Leydig cell. It can either diffuse into the Sertoli cells, or it can pass through the blood. We will see this in a few minutes. Why is it important for testosterone to travel through the blood? does it have an inhibitory effect on the hormonal secretions? We will see this in a few minutes. Now, the important thing at this moment is to know that testosterone that is synthesized by the Leydig cells is either passing through the blood to act by endocrine activity, or it can travel into a neighboring cell, which is the Sertoli cells, by paracrine activity to act inside of the Sertoli cells. It can pass through the membrane. It can be transformed into dihydrotestosterone by the activity of the reductase. Also, it can lead to the formation of estradiol by the activity of the aromatase, or it can pass through the Sertoli cells to reach to the lumen, where it binds to the androgen binding proteins and gets concentrated inside of the lumen. Now, what are the major roles of testosterone? The testosterone is very important because it participates in the activation of what we call the hypophyseal testicular axis. What does it mean? The testosterone will act like an intermediate molecule or a relation molecule between the hypophyseal axis, the hypothalamus anterior pituitary axis, GnRH that stimulates FSH and H. So this acts of hormonal control, as well as the testicular axis. So it will act as an intermediate to let these two axes communicate, the hypophyseal axis and the testicular axis. Now, also it's important for the differentiation of the male genital organs. We saw this in the chapter 3. We mentioned that testosterone is very important for the formation of the male genital organs. It's important for the masculinization of the urogenital sinus, as well as the external genitalia, especially the DHT form of the testosterone. It's important because it will acquire and maintain the secondary characteristics and male behavior, especially after puberty, from puberty till the end. It's very important because testosterone as well as FSH, which are both acting on Sertoli cells, will ensure that this Sertoli cell will maintain the integrity and will allow the development, or will allow briefly spermatogenesis. So because of the response to testosterone and to FSH together, so the as we mentioned, Sertoli cells, they have receptors for FSH as well as receptors for testosterone. Both these molecules, FSH and testosterone, will help Sertoli cells provide a good environment and the right molecules for the development of the spermatogonial cells into the formation of sperm cells. Therefore, they are important therefore, testosterone is important to initiate and maintain the spermatogenesis. We'll start with the first of the functions, which is the neuroendocrine control of testicular function. So as I mentioned, the central nervous system that is controlled by the hypothalamo-hypophyseal axis. So the hypothalamus secreting GnRH, the gonadotropin releasing hormone that is produced by the neurosecretory cells of the hypothalamus, will be released and will come in contact with capillaries of the hypothalamic hypophyseal portal system. So this system here is highly capillarized in order to allow a rapid transport of the GnRH to the anterior pituitary, and it will stimulate the anterior pituitary to secrete the so we call it the adeno hypophysis, or we call it the anterior pituitary. It's the same thing. It will trigger them. It will rapidly transfer GnRH to this anterior pituitary, so that this one can express the pituitary gonadotropins, which are the FSH and the LH. So FSH, which is the follicle stimulating hormone, and the LH, which is luteinizing hormone. Therefore, the important thing is that these two systems that are here, the hypophyseal axis, will be communicating with the reproductive system, because this FSH will act on the testes. Particularly, they will act on the Sertoli cells, and they will regulate the secretions of the Sertoli cells, in addition to testosterone that is also acting on these Sertoli cells. Now, these GnRH and LH and FSH, they have a pulsatile mode of release, and they are, in their turn, they will stimulate the gonads. So the GnRH stimulated the release of LH and FSH, and we mentioned that LH and FSH, we call them the gonadotropin glycoproteins because they act on the gonads. So LH and FSH, both of these molecules will be released to stimulate the gonads. Now, FSH will be acting on the Sertoli cells. However, LH will be acting on the Leydig cells. This is very important to know at this level. Therefore, FSH will act on Sertoli cells and LH will act on the Leydig cells. In order for FSH to act on the Sertoli cells, of course, Sertoli cells must have receptors for FSH, and these receptors are the cell surface receptors. The same applies for LH. For LH to act on the Leydig cells must possess receptors for the cell surface receptors for the LH. Now, these gonadotropins that act on the gonads will stimulate these gonads to secrete steroid hormones. Okay, so they will, LH, for example, will stimulate the Leydig cells to secrete the testosterone, and the testosterone will be also going into the Sertoli cells and will be transformed into estradiol. And this transformation will not be possible if there is no FSH, because FSH, when it binds to the receptor, it triggers the expression of the aromatase enzyme that is needed for testosterone to be transformed into estradiol. So this is very important. This is why we say that LH and FSH are important for the secretions of testosterone and estradiol by the gonads, as well as they will stimulate the secretions of protein factors like inhibin and activin. So already we mentioned that inhibin and activin are secreted by the Sertoli cells. We mentioned that inhibin inhibits and activin activates. We are going to see this all in few minutes. Now, steroid hormones and inhibin, they regulate the synthesis and secretions of pituitary and hypothalamic hormones by exerting a feedback regulation that is negative feedback inhibition. What does this mean? This means that this inhibin that is secreted by the Sertoli cells can act back on the anterior pituitary to inhibit the secretions of FSH. So you can see that inhibin is inhibiting the secretion of FSH by acting in a negative feedback inhibition. So it can go into the blood, of course, to travel through the blood to reach to the anterior pituitary where it can inhibit the secretion of the FSH. The activin, of course, it works in the opposite way. And also the testosterone, you can see here, testosterone is traveling through the blood, and it can also exert a negative feedback inhibition on the GnRH secretion. Now, how is the spermatogenesis controlled? How is the hormonal control of spermatogenesis? Now, the spermatogenesis is controlled by the action of the gonadotropic glycoproteins, so FSH and LH. Now, as I mentioned, Leydig cells, they have receptors for LH. So once the LH molecule binds to the cell surface receptor, remember the cell surface receptor mechanisms of action. We mentioned that we have the G protein. G protein that is coupled to the receptor will activate the adenyl cyclase that will produce cyclic AMP second messengers. Cyclic AMP second messengers messengers activate protein kinase A. Protein kinase A will activate the synthesis of new proteins. These new proteins include, they are not only, they include the enzymes that are important for the conversion of cholesterol to testosterone, which are the steroidogenesis enzymes. Okay, so they are synthesized because of LH binding to the receptor. So this is very important. So the spermatogenesis, it depends on the action of gonadotropic hormones FSH and LH, because there are receptors for these two. Now, this testosterone will rapidly get into the blood. It can get into the blood and travel to exert a negative feedback, as we mentioned, but also it can diffuse locally into a neighboring cell by paracrine secretion, and it will go and bind to intracellular receptors. So it can diffuse through the membrane because it is a steroid hormone. It can diffuse and act inside of the cell. It can, so therefore, bind to intracellular receptor, and it can go to the nucleus and activate the synthesis of new proteins. Okay, moreover, it can pass into the lumen and bind the ABP after being responsible for its synthesis. So when testosterone enters inside of the Sertoli cells, it can trigger the synthesis of new proteins, including inhibin, including androgen binding proteins. Okay, so it can stimulate the synthesis of so many proteins. And one of these important proteins is ABP, androgen binding proteins, that can bind to the testosterone and concentrated in the lumen of the seminiferous tubule. On the other hand, the passage of testosterone from the Leydig cells to the Sertoli cell will not act alone. Testosterone is acting in synergy with the FSH. So at the same time that testosterone has diffused into the Sertoli cells, FSH that came through the blood and reached to the Sertoli cells, it can bind to the cell surface receptor. So it will bind to receptors, especially where are these present? They are present at the basal compartment. This is very important. The Leydig cell is not polarized cell. ما عندها basal and apical compartment. بينما Sertoli cells, it has basal and apical. وين موجودين the FSH receptors? The FSH receptors, they are present at the basal plasma membrane of Sertoli cells. So once it binds to the cell surface receptor, the same as the LH receptor, it will trigger the signaling transduction, leading to the activation of protein kinases that will lead to the RNA synthesis and then translation of proteins, including APP and inhibin. Therefore, both testosterone and FSH, both of them, they will work in synergy together in order to reach a better response to synthesize inhibin, to synthesize ABP, which is the androgen binding proteins. And this androgen binding proteins can bind both the dihydrotestosterone and the testosterone in order to allow their concentration inside of the lumen of the seminiferous tubule, and this is very important for the continuity of the spermatogenesis, particularly the spermiogenesis, which is the formation of the sperm cell at the end. Now, we mentioned that activin and inhibin, they can exert feedback inhibition mechanisms. What are these feedback inhibitions? Now, in this figure here in front of you, you have the GnRH that was secreted by the hypothalamus to act on the anterior pituitary and to trigger them to release the LH and the FSH. Now, when this mechanism is done, at a certain time, we need to turn it off. We need to slow it down. Therefore, you can see that inhibin that was produced by the Sertoli cells can act by negative feedback. So inhibin B, you can see here, you have inhibin B. It acts by negative feedback to stop or to inhibit the synthesis of the or the release of FSH. Okay, so inhibin produced by Sertoli cells in response to FSH exerts a negative feedback on the pituitary gland to reduce the serum FSH amount. The opposite is done by activin. So activin will exert a positive feedback where it can activate the FSH release. Now, one would see here that you have the Leydig cells that have also inhibin. There are two types of inhibin. There is the type inhibin, we call it inhibin A and inhibin B. The inhibin B is the one that is more active in the males, in the human males. So this one is the one that is secreted by Sertoli cells and that acts by inhibiting the FSH. Is the more potent. It's more, you can see it, it's put in bold because it's the more active one. So inhibin B is the one that inhibits FSH secretion, and activin is the one that activates the FSH secretion. The other feedback mechanism is exerted by the testosterone. So testosterone that can travel, as we mentioned, through the blood. So either it can go to the neighboring Sertoli cell or it can go through the blood. It can exert a negative feedback inhibition on LH secretion and also on GnRH secretion. And this regulation loop is important because it can control the Sertolian function. Okay, so it can stop indirectly the GnRH secretion and thus the FSH secretion. So it can control both the Leydig and the Sertoli cells. Now we will talk about the interactions that exist between each of these different cells. So interaction between Sertoli and Leydig, interaction between the peritubular cells, which are the myoid cells, and interaction between the germ cells and the Sertoli cells. What are these different interactions? Now, the first important thing is that the molecules that are secreted between all of these different Leydig, Sertoli, germ cells, and peritubular cells, these molecules, they are either secreted by a paracrine or by an endocrine way. And of course, now if we are talking only about these four, they are very neighboring to each other. So it could be, it's either by paracrine or sometimes autocrine, where the cell can respond to itself. For example, the activin and inhibin that acts on Sertoli cells themselves. Now we start with the first one, which is Sertoli and Leydig cells interaction. How are Sertoli cells controlled? Are they controlled by molecules from Leydig cells, from pituitary? Actually, it's both. They are controlled by the FSH, which is released by the anterior pituitary, and by testosterone that is given to it from the Leydig cells. Therefore, for this Sertoli cell to be able to respond to these two molecules, first it has a cell surface receptor that allows it to respond to the FSH secreted by the anterior pituitary, and an intracellular receptor that is ready to bind to testosterone that is secreted by the Leydig cells. Therefore, this testosterone will act as a paracrine factor that will control the functioning of Sertoli cells and will play a role as a communication or a connection between Leydig cells and Sertoli cells, and after that germ cells, because this testosterone will not reach to the germ cells. Germ cells will not be able to respond to testosterone, so it will be going from Leydig cells to Sertoli cells, and what happens inside the Sertoli cells in response to testosterone will be acting on the germ cells. Okay, so we will see this in a few minutes. Now, why is this the case? Why is the germ cells not responding to testosterone? It's as simple as this. For any cell to respond to any molecule, it needs to have receptors for them. Germ cells do not have receptors for androgens. So germ cells will not respond to testosterone. طب كيف testosterone as a steroid hormone is allowing the spermatogenesis? It's allowing it via Sertoli cells. It will act on Sertoli cells and will trigger Sertoli cells to secrete molecules such as, for example, you can see here you have the IGF-1 and the TGF-1, and you have also the activin, okay, and you have the inhibin. All of these molecules that are secreted by the Sertoli cells are the ones that will act on the germ cells. So here you have the germ cells and here you have Sertoli cells. So these Sertoli cells, they will respond to testosterone and they will secrete inhibin, they will secrete activin, they will secrete the IGF and TGF, and by secreting this molecule IGF, TGF, activin, and inhibin, they will be controlling the germ cells. Okay, so again, germ cells do not have receptors for testosterone or for androgens, and thus cannot respond to them. So the testosterone that is produced by the Leydig cells is modulated locally by factors that are synthesized by Sertoli cells. So Sertoli cells will respond to testosterone and synthesize growth factors such as insulin growth factor 1, and it will secrete also inhibin. Inhibin and activin, they act differently on germ cells. Okay, so it's very important at this time to pay attention to this information. In the table, when I was talking about the functions, so table 4.1, when we were talking about the inhibin and activin, we mentioned that the inhibin, it as you can see here, so inhibin is inhibiting the stimulation or the proliferation of spermatogonia, and we mentioned that activin activates the spermatogonia. So these two molecules that were synthesized by the Sertoli cells will either activate or inhibit the germ cells. So this is the way it will respond to the testosterone. So it's via the Sertoli cells. Now, very important thing is that in addition to the secretion of inhibin and activin that acts on germ cells, these Sertoli cells, by secreting activin and inhibin, they will also act on themselves. Actually, what happens here, so you can see these Sertoli cells that secreted inhibin and activin, these two molecules will go and act again on the Leydig cells. But here it's the opposite way. You can see activin now is inhibiting Leydig cells. However, inhibin is positively stimulating the Leydig cells. Stimulating them for what? It is stimulating them to synthesize more of the testosterone, or inhibiting them from synthesizing less of the testosterone. So it's like a control way between Sertoli cells and Leydig cells. If Leydig cells synthesize a big amount of testosterone, Sertoli cells can respond to this big amount of testosterone by synthesizing huge amounts of activin and inhibin. Activin and inhibin will help these germ cells to develop because activin activates and inhibin inhibit. However, when they act on the Leydig cells, they act differently. So activin will be acting locally, so they will be binding to the cell to inhibit, so they will act on Leydig cells. Activin will be inhibiting steroidogenesis, whereas the inhibin will be activating or stimulating the steroidogenesis in addition to IGF-1. Therefore, IGF-1 and inhibin stimulate steroidogenesis, while TGF-beta and activin will inhibit steroidogenesis. And I repeat, steroidogenesis is the formation of testosterone inside of the Leydig cells. Now, at the same time, this IGF here, that is excreted at the basal compartment. So here, this is the basal compartment of the Sertoli cell. Now, this IGF that is excreted at the basal compartment of the Sertoli cells, they can also bind to specific receptors that are in the Leydig cells, and they will be also actually making the IGF will be binding to receptors on the Leydig cells and will be stimulating these Leydig cells to synthesize more receptors for LH. And when they synthesize more receptors for LH, thus they can synthesize more of the androgens. So they will stimulate steroidogenesis. Now we move to the second type of interaction, which is the Sertoli peritubular cells interaction. Now, the peritubular cells are these cells, the myoid cells. They are present, as we mentioned, in the lamina propria. Now, they secrete various factors that regulates the activity of the serum. They express large numbers of androgen receptors, so they can respond to androgens. Okay, so they can respond to testosterone, unlike the germ cells. And when they are stimulated by testosterone, so positive means stimulation, negative means inhibition. When they are stimulated by testosterone, they can secrete a PMODS factor, which is the peritubular modulating Sertoli cell function. And this molecule will act on Sertoli cells and will again stimulate it to synthesize androgen binding protein, transferrin, and inhibin. Another interaction is the Sertoli germ cell interaction. Now, we mentioned previously that germ cells will respond to Sertoli cells because of the molecules that are secreted. So I mentioned in this figure here that the germ cells will respond to the activin and inhibin that are, and IGF and TGF that are secreted by the Sertoli cells. Now, how is this? How do these germ cells respond? What do they secrete? They will secrete the EGF and the NGF, so the epidermal growth factor and the new nerve growth factors are two growth factors secreted by the germ cells. And they will, which type of germ cells, particularly the primary spermatocytes and the round spermatid, will secrete these molecules in order to regulate the Sertolian function. At the same time, when these Sertoli cells, they have phagocytosed the residual body, this will stimulate them to secrete the interleukin-1 and interleukin-6. And I repeat, the interleukin-1 activates the DNA replication, interleukin-6 inhibits it in spermatogonia and primary spermatocytes. Okay, so therefore there is a reciprocal relation. They respond to each other. Sertoli cells, they phagocytose the residual body. This will trigger them to synthesize interleukin-1 and interleukin-6 that will control other developing spermatogonial cells. They will control their replication of DNA. وبنفس الوقت when the primary spermatocytes and the round spermatids are formed, they will secrete the epidermal growth factor and the NGF, which are very important factors to stimulate Sertoli cells to secrete again these hormones. So it's like as if there is a communication and we are responding to your molecules, so they secrete to each other so that they can communicate via these molecules. Now I move to a new part in this chapter, which is the sperm transport. Now we mentioned that the sperms, when they are formed in a by spermiogenesis, or the differentiation of sperm. Now, very important thing here, the sperm has acquired a different shape, is now no more like a spermatid. It has a shape of a sperm. It is more likely the ant form. A little bit of changes will occur with respect to its structure, but the major structure, it has acquired it inside of the seminiferous tubule. However, after spermatogenesis, when the sperm is undergoing spermiogenesis, which is the cytodifferentiation, it will lead to the formation of a sperm cell that is, we can say it's a mature spermatozoa. However, it's not a 100% mature. This is why I prefer not to use the term a mature spermatozoa without saying that it's not motile and it's not a fertilizing sperm. So the sperm that is released in the lumen of the seminiferous tubule is not a completely mature spermatozoa. This is very important. It's partially mature. It's not completely mature لأنه it needs to go into the epididymis to acquire more maturation. And again, when it passes into the female reproductive system, it will acquire further properties that will make it a fertilizing sperm. لأنه وقت يكون released in the seminiferous tubule, it's not a fertilizing sperm cell. The release into the lumen of the seminiferous tubule is what we call the spermiation. It's when these sperm cells are detached from the Sertoli cells and they are released into the lumen of the seminiferous tubule. How are these sperms transported? First of all, they are transported from the seminiferous tubule to the straight seminiferous tubules, or the straight ductules here, and then they move to the mediastinum testes or the rete testes that we mentioned this place as the body of Highmore, where they reach to the number 4, the efferent ductules, so that they can reach finally to the epididymis. Therefore, the movement all over the seminiferous tubule to the straight tubules, then to the mediastinum, the rete testes, then to the efferent ductules, all of this is triggered by peristaltic contractions of the myoid cells, as well as by the fluid that is present with the sperm cells, the fluid pressure, the intraluminar pressure that is triggering them, because at this moment, the sperm in the lumen is not motile sperm. Now they will travel into the epididymis. The epididymis is a 5 meters coiled epididymis duct, and during their travel into the epididymis, they will acquire motility. Okay, so this is very important. They will start first by the head of the epididymis, the body of the epididymis, so they will start going into the head, the body, and then they will be stored in the tail of the epididymis. They will be stored there so that they will be released at later part. So the tail is used for the storage of the sperm. However, the exchanges that exist between the sperm cells and the epididymal cells, the cells of the epididymis, will take place in the head and in the body. And this exchange will make sure that during the passage through this part, it will acquire maturation. Now, they will be stored in the tail of the epididymis. In the absence of ejaculation, they will be deteriorated as they age, and they will be lysed and phagocytosed. Okay, so we get rid of the sperms if after a time they are not released. Now, how is the wall of the epididymis? This epididymis tube is, it has a wall, it has a epithelium that is lining it. The wall of the epididymis is surrounded by a the pseudostratified columnar seroselated epithelium. So we have, first of all, the wall that has this smooth muscle cells that are important to provide contraction, and below this smooth muscle cells, there are the pseudostratified epithelium. Now, why do we call it pseudostratified? Because actually it's not a stratification. It's not 2 layers of cells. It's 2 types of cells. There are the basal cells, cells that are short cells toward the basement membrane, and there are columnar cells which are going from down till the top. And these columnar cells, they possess what we call as stereocilia, which are similar to the microvilli, and they provide a big surface for exchange of molecules. Okay, so of course, stereocilia should be toward the lumen of the epidermis. So again, there are a smooth muscle layer, and then a pseudostratified columnar stereociliated epithelium made of two types of cells. The principal cells, which are the columnar cells that are ciliated, they have stereocilia, and there are the basal cells that are toward the basement membrane. Now, because of the contraction of this smooth muscle layer, these sperms, they can move by peristaltic contraction, and they will reach to, as I mentioned, to the tail, where they will be stored. How is the sperm cells, how can they survive? Now, the sperm cell can survive several weeks in the male duct system, and they survive only 2 to 3 days in the female reproductive system. This sperm, before leaving the epididymis, they will be acquiring special types of proteins on their head, on their head part, and these proteins will provide them with a rigid form, so that they can tolerate the passage into the female reproductive system. And once they are in the female tract, they should lose this molecule that they have acquired in the male reproductive system, so that they become fertilizing. Okay, why I'm mentioning this? Because this is very important. The capacitation is one of the steps that are very important for the sperm cell to be able to become a fertilizing sperm. Now, the sperm, after spermiation, it's morphologically differentiated. It's leaving the testes, it's leaving the seminiferous tubule and entering the epididymis. They are non-motile and they are non-fertile. They need to acquire maturation, a functional maturation, so maybe shape-wise or morphologically they are mature, but functionally they are not mature yet. So they need to acquire functional maturation so that they become motile and fertile. And this maturation occurs as different steps. So as they transit through the epidermis, they will undergo changes so that they become able to recognize and fertilize the oocyte in case there is a sexual intercourse and fertilization. Therefore, it will acquire, by passing into the epidermis, it will acquire motility, and it will, after ejaculation, it will be activated. And when it is going into the female reproductive system, it will furthermore undergo capacitation, which means removal of what they have acquired in the epidermis. The proteins that were bound to its head to provide rigidity will be lost during ascension to the ovary. This is capacitation, which is very important for the fertilization. And near the oocyte, when it reaches to the oocyte, there is acrosomal reaction. Faizan, I'm mentioning all of these steps that will be mentioned in chapter 6 to tell you that this sperm, even by the passage in the epididymis, it's not 100% fertilizing. Okay, so the sperm will undergo maturation as it moves through the epididymis. It will acquire motility and fertility. However, its full fertilizing capacity is not acquired in the male reproductive system. In the male reproductive system, it will acquire epididymal maturation. However, in the female reproductive system, إذا صار في sexual intercourse and these sperm cells were ejaculated in the female reproductive system, this sperm will undergo capacitation and acrosomal reaction, and this will make them fully fertilizing, and this will be discussed in the chapter 6. What are the functions of the epididymal canal? Therefore, the epididymal canal is important to, first of all, transport the sperm cells. It will provide secretions. It will reabsorb molecules coming with the semen, with the sperm cells, or with the tubular fluid. tubular fluid اللي جاي مع sperm في بقلبه androgen binding proteins في بقلبه androgens. So many molecules are coming with the sperm cells. There will be reabsorptions, metabolism, and maturation of the sperm. We start with the first one, which is the transport of the spermatozoa. Now, the transport of spermatozoa occurs via extra-testicular ducts. So the movement, first of all, from the epidermis, they will move to the vas deferens, or we call it the ductus deferens. So here, from the tail, they move to the vas deferens. Okay, so from the tail, they move through the vas deferens. Then they will reach to the ampulla. Then from the ampulla, they move to the ejaculatory duct. Then they move to the urethra so that they are released outside of the body. Okay, now the progression through all of these, it depends on two things. It depends on what we call as the intraluminary pressure, the pressure inside of these ducts, and the contraction of the tunica albuginea, which is present covering all of this. So it is covering all of these ducts. The tunica albuginea will provide the contraction. Once they reach into the tail of the epidermis, they need to pass into the vas deferens, and they move through the vas deferens, as we mentioned. They reach to the ampulla. Here, in the ejaculatory ducts, they get mixed with secretions from the seminal vesicle. Then they continue, they get mixed with secretions from the prostate gland. They continue, they get secretions from the bulbourethral gland, which is the Cooper gland, and they reach to the penis, and they are ejaculated outside, mixed with all of these secretions from these accessory glands. And this is what we call the semen. So the semen is this mixture of all of these secretions. Now, secretions that are done in the epididymal epithelium include the ions, so the epididymal secretions here in the epidermis, the sperm cells that will be leaving the tail and moving into the vas deferens are, they have secretions like ions, potassium, and phosphorus. They have small organic molecules. They have proteins and glycoproteins. All of these are very important because they will provide motility for the sperm. They will start to help it become later on a fertile sperm cell. And these glycoproteins that will be assembled on the head of the sperm cells will be later used for the fixation to the zona pellucida at the time of the fertilization, as well as they will be protecting the sperm from having an acrosomal reaction that is premature. يعني it will allow the acrosomal reaction to occur, but not in a premature way. It will allow it to occur only when they reach to the female reproductive system. The reabsorption occurs also in the epidermis. The epidermis will reabsorb 90% of the fluid coming from the testes, as well as ions, proteins like androgen binding proteins and transferrin will all be absorbed by the epidermal epithelium, and this will lead to the concentration of the sperm cells inside of the epidermis. It will also, this reabsorption of the fluid will stimulate the epidermal cells to undergo glycolysis and start metabolizing fatty acids, and will also allow them to start synthesizing prostaglandins and convert testosterone to dihydrotestosterone. And all of these are very important because they will help also, again, the sperm cells undergo maturation. And this part of the maturation that is undergoing inside of the epidermal tube. What is this maturation? Now, all of these, what I mentioned here, whether the reabsorption, the concentration of sperms, all the secretions, all of these will contribute to the maturation of sperm cells. Remember, we mentioned that when the residual body is removed, there will be a cytoplasmic droplet that was remained in the sperm cell. And I told you last time that this cytoplasmic droplet will be lost during epididymal transit. This is one of the maturation of the sperm cell. Another maturation is that the nucleus will undergo more compaction or condensation. The chromatin will form disulfide bonds more and more between the cysteine of the protamines. This already started in the testes, but it will be more prominent, more important at the level of the epididymis, so that the nucleus undergo its final maturation. This is the last shape of the nucleus. It will not undergo more compaction later on. The acrosomal enzymes, the hyaluronidase and the acrosine that are present in the acrosomal cap, they will be neutralized by the epididymal secretions. So the epididymis is secreting special types of molecules to neutralize the acrosomal enzymes. Why is this the case? These proteins that will be surrounding the head of the sperm cell will prevent, in case there is a leakage of the acrosome, leakage from or a lysis of the sperm cells, so that these acrosomal enzymes does not cause damage to the male and female genital tract. Yani in case صار في acrosomal release, premature acrosomal release, that is not its time to be released, the presence of these epididymal secretions will prevent any damage that can be caused by these acrosomal enzymes. لأنه هن hydrolytic enzymes, they are lytic enzymes. Also, the mitochondrial helix will complete its placement during the epididymal transit into the neck. So they will acquire their final location, and there will be an increase in glycolysis and a pH increase in the pH because of metabolic reactions. And all of these, the structural, biochemical, and metabolic changes that occurred are all will be involving the acrosome, will be involving the nucleus and the plasma membrane. We start with the first one, which is the decapacitation. Now, I mentioned that they will acquire proteins that will protect the enzymes from undergoing the lysis. This is called decapacitation. Now, the plasma membrane of the sperm cell will have proteins that will be fixed on it, and these proteins come from the epididymis. So they are epididymal proteins. They will remove the testicular proteins that came with the sperm cells from the seminiferous testes and will replace them with epididymal proteins. These epididymal proteins will be protecting and stabilizing the sperm and protecting it from undergoing a premature acrosomal reaction. هيدا بالنسبة للبروتينز that are on the membrane of the sperm cell. What about the lipid composition? كمان the lipids will be changed. There will be an increase in the cholesterols at the level of the membrane of the sperms and a decrease in phospholipids. When we decrease phospholipids and increase cholesterol, we are decreasing the fluidity of the sperm. عم نعمله more rigid so that it can tolerate traveling long distances. وقتها يوصل على الأج على الoocytes وبده يعمل fertilization، هونيك بنرجع بنعمله more fluid to allow the fusion of the two membrane. هيدا بنسميها decapacitation، نحكي عنها بchapter 6. At this moment, the sperm is decapacitated. Its membrane is stabilized. How is it stabilized? It's stabilized by changing its lipid composition as well as by changing its protein composition. The functional maturation also includes the acquisition of unidirectional motility. Now it can move. It has a capacity that to move in a straight linear progression. So there are a flagellar curvature of small amplitude now. So the movement is possible because of what? What make it acquire a unidirectional motility? There will be disulfide bridges that will be formed between the tail proteins, and this will make the flagella more rigid and will increase the production of cyclic AMP at the level of the tail, and all of this will help it move in a straight linear progression. Also, there will be appearance of receptors to the zona pellucida at the level of the acrosome, as well as receptors of the oocyte plasma membrane in the post-acrosomal region. This part will be talked about in details in the chapter 6. I will talk a little bit about it now. So the zona pellucida, which is it's a translucid, or it's an acellular part that is surrounding the oocyte. has sperm cell يقدر يفوت على oocyte يعمل fertilization. It's crossing. It needs to cross follicular cells as well as the zona pellucida. Okay, so in order to cross the zona pellucida, أول شيء بدها تعمله, it needs to bind to it. So ضروري هالسبرم cell يصير عنده receptors لهالزونا pellucida. So هون it will acquire the receptors for zona pellucida at the level of the acrosome. وتاني شغلة very important, it will acquire receptors. خلصت الزونا pellucida وصارت in contact with the oocyte plasma membrane. كمان بده يكون في عندها receptors for the plasma membrane. وين بيكونوا موجودين؟ بريجن من بعد ما يصير في acrosomal reaction ويبطل فيه الأكروزوم. هو it's a bag that will release the enzymes. بعد الأكروزومال reaction بيبقى شيء اسمه post acrosomal region, بيكون فيها receptors so that the sperm cell can bind to the plasma membrane. All of this will be seen, of course, in details in chapter 6. I will move to the last part of this chapter, which is about the acrosomal, the accessory gland, sex glands. So we mentioned there are 3 types of accessory sex glands that exist in the male. So there are the seminal vesicles, there is the prostate gland, and the bulbourethral gland. Now, these secretions, these glands will secrete fluids that will mix with the sperm cells to form what we call the semen. Okay, so السائل المنوي. So the semen is a liquid that is very important because it will provide viability for the sperm cells inside of the female reproductive system. Now, the 2 to 6 ml ejaculate from the penis contains approximately 40 to 250 millions of sperm cells. 60% of this semen is given by seminal fluid secreted by the seminal vesicle. 30% of the semen is prostate fluids by the prostate gland, and 1% is the mucus secreted by the bulbourethral gland. Okay, and 9% will be the sperm cells. Then 9 to 10% will be the sperm cells, in addition to so many molecules. We start with the first gland, which is the seminal vesicles. These are like sacs that are attached to the vas deferens near the base of the bladder. The important thing is that they secrete high concentrations of fructose. Fructose will provide ATP energy for the sperm to be motile. And seminal vesicles also secrete prostaglandins. Prostaglandins are important because they will trigger the contractions of smooth muscles in both the male and the female reproductive system. يعني وقت يكونوا they are moving in the male reproductive system to get ejaculated outside of the body, prostaglandin will trigger smooth muscle contraction in the male reproductive system. Once they are ejaculated inside of the female reproductive system, also prostaglandin coming with the sperms will trigger the contraction of the female reproductive system. All of this will make the functional sperm a motile sperm. It will help it to be motile. The other gland is the prostate gland. The prostate gland will secrete a milky fluid that is neutralizing fluid. It will neutralize the acidity of the urethra بالمale. هالميل وقت the sperms are ejaculated, they pass through the urethra that is acidic. To neutralize the acidity of the urethra, the milky fluid secreted by the prostate gland will be making sure to neutralize the acidity of urethra, as well as in the female reproductive system, in the vagina especially. The vagina is highly acidic. كمان it will contribute to the neutralization. So this prostate gland that you can see here, now this prostate gland is a walnut size gland that is located below the urinary bladder in front of the rectum. The last gland is the bulbourethral gland أو بنسميها Cooper gland. There are two, so bulbourethral glands, Cooper glands, two from both sides. These glands, they secrete a clear and slippery fluid. This fluid, like a mucus fluid, will neutralize the acidity from urine and urethra, but also in urethra, but also will provide lubrication. It is a pea-sized gland that is located on the sides of the urethra just below the prostate gland, and it's very important because it is important for lubrication. Okay, so that's it. That's all for this chapter. So I will go over the tables and to recapitulate, and then I will take all your questions. So this table inside your book summarizes all the types of the different components of the male reproductive system. The testes, which are pair of glands located in the scrotum, their function is to produce sperms and to secrete testosterone. The epidermis and the ductus deferens, also they are paired, and each one of the testes, they have attached to them one epidermis. Epidermis and ductus deferens is important because it will provide the maturation and the motility of the sperm cell. It will concentrate and store the sperms and will serve as an exit route from the testes to the outside of the body for the ejaculation. The three glands, the seminal vesicles, prostate gland, and bulbourethral glands, provides with the semen, the fluid that is given with the sperm cells. Seminal vesicles are pairs and bulbourethral glands are pairs. However, they have only one prostate gland. The seminal vesicles, they will empty into the ductus deferens. The prostate gland will surround the urethra, and the bulbourethral gland will empty into the urethra. Seminal vesicles provide fructose for the energy and prostaglandin for contractions. Prostate gland will provide a milky fluid for neutralizing the acidity of vaginal secretions. Bulbourethral gland, both the mucus is for neutralizing, but also for lubrication, especially during the sexual intercourse. Finally, we move to the major reproductive hormones in males. GnRH, secreted by the hypothalamus, acts on anterior pituitary to stimulate it to secrete LH and FSH gonadotropins. The LH acts on the Leydig cells because they have receptors for them. They have cell surface receptors for LH, and once LH binds to Leydig cells, it will trigger a signal transduction that will lead to the synthesis of enzymes related to the synthesis of testosterone from cholesterol. LH is secreted by the anterior pituitary. Now, FSH, secreted by the anterior pituitary, acts on the Sertoli cells, on cell surface receptors of the Sertoli cells. Also, this FSH will trigger signaling transduction, leading to the synthesis of enzymes, leading to the synthesis of aromatase and reductase, but also to the synthesis of ABP, inhibin, and so many other interleukin-1, interleukin-6. So all the molecules, IGF, all the molecules secreted by the Sertoli cells are stimulated by the FSH, and all of this will support spermatogenesis. Testosterone, which is a very important and active androgen, secreted by the interstitial cells, the Leydig cells particularly, as well as it can be synthesized by other types of glands. Testosterone can act on the testes, as well as it acts not only on the testes, it can act on so many other tissues, on the body tissues. And this testosterone, therefore, it can help in the development and maintenance of reproductive organs, as well as to support spermatogenesis and to help develop and maintain secondary sexual characteristics. Now, it can act on anterior pituitary and hypothalamus as a negative feedback regulation or feedback inhibition by inhibiting the secretions of GnRH, LH, and FSH. Okay, so all of this is about the chapter 4 for the male reproductive system. Now I will take your questions.